Good morning students, myself V.T.BIOLOGY, PGT Biology from PG Senior Secondary School. I am here with my next topic, that is Kingdom Monera for class 11th. So in this kingdom we discuss about the main characteristic feature of Monerans and after that examples. So especially according to 5 kingdom system of classification, the organisms are classified into 5 kingdom by R.H. Whittaker, that is the Monera, after that Protista, after that Fungi, Plantae and then Animalia. So in increasing order, because in case of kingdom Monera, all the primitive plus unicellular organisms are kept by R.H. Whittaker. After that Protistans are well developed than Monerans. After fungi more developed than protist. After that plants more developed than fungus, and last Animalia means advanced and well developed comparison than Monerans, Protistans, fungi and Plantae. So in case of Monera, Monera includes all prokaryotes, means all the prokaryotic organisms are kept in under Monera because their nucleus is not well developed and they are unicellular, especially bacteria and cyanobacteria. So Monera includes all prokaryotes such as bacteria, mycoplasma, actinomycetes and blue-green algae, that is cyanobacteria. So it's mean that the Monera are specially allotted for all the prokaryotic and unicellular organisms because their nucleus is not well developed, their nucleus is in primitive stage. Now about the characteristic features of Monerans. So now about characteristic features of kingdom Monera. So in case of characteristic features, the first number is all the simplest or most primitive unicellular organisms are kept in under Monera. So Monera includes all the simplest and most primitive and unicellular organisms in which the nucleus is not well developed, the nucleus is in primitive stage. Primitive means not well developed. Next number, in case of Monerans, cell wall is present. We already know that in case of plant cell wall is present, but their chemical composition is different, that is made up of cellulose. In case of fungal cell wall is made up of chitin. While in case of Monerans, especially we are talking about bacterial cell. In bacterial cell wall is present and their chemical composition is different from the cell wall of plant cell wall of fungal cell, because the cell wall of bacterial cell is made up of peptidoglycan or murein. It is made up of peptidoglycan or murein, means the cell wall of Monerans are not made up of cellulose, means cellulose is absent in case of cell wall of bacterial cell, especially in case of Monerans. So their cell wall is made up of peptidoglycan or murein. This is very important for 1 marks, means the bacterial cell wall is made up of, so bacterial cell wall is made up of peptidoglycan or murein. Next thing, in Monerans, means especially in case of cyanobacteria or bacteria, cell membrane bound organelles like mitochondria, like Golgi body, like endoplasmic reticulum, like vacuole, these all type of organelles are absent in Monerans. In case of Monerans, all the membrane bound organelles are absent, means no any membrane bound organelles are present in bacterial cell as well as in case of blue-green algae. Next, there are various type of nutrition occur in case of Monerans, that is especially saprophytic. Saprophytic means they exist, means they depend on dead and decaying organic matter for their food material. So organisms which depend on dead and decaying organic matter, they are called saprophytes, and their mode of nutrition is called saprotrophic or saprophytic mode of nutrition. After that, some are parasitic, means they are depend on living body, that is the plant body or animal body. After that, chemoautotropic, means synthesize their food with the help of some certain chemicals. After that, photoautotropic, depend on light. And symbiotic, means some bacteria like rhizomium present in root nodule of legume crops, and they show symbiosis between that legume crops and environment because they fix nitrogen from environment and provide to the plants. After that, DNA is naked. DNA is naked means generally in case of eukaryotic cell, DNA is present inside the nucleus. While in case of monerans, nucleus is not well developed, means the membrane, that is nuclear membrane, is not present in the nucleus of the monerans. So DNA is naked, it is not surrounded by any membrane like nuclear membrane. Next thing, it lies inside the cytoplasm. Cytoplasm is commonly present in all type of cell. So it lies inside the cytoplasm in coiled form. Means suppose that this is a bacterial cell, and inside bacterial cell cytoplasm is present, and in cytoplasm DNA is present in this type. Means this is a coiling type structure, means DNA is present in coiled form like this, and it is present on the place of nuclear. So this is called nucleide. So in bacterial cell or in case of monerans, nucleide DNA is present. Next number, flagella. Flagella is a thread-like structure, and in case of monerans, flagella helps in the movement of the monerans from one place to another place. And flagella is made up of a special type of protein, that is flagellin. So flagella is made up of flagellin, and flagellin is a type of protein. So in case of monerans, flagella is helpful in the locomotion, means through flagella they can move from one place to other place, and flagella is a thread-like structure. Reproduction. In case of monerans, especially asexual method of reproduction is present because there are no any gametes are formed, so gamete formation not occur in case of monerans, and due to absence of gametes, sexual reproduction is not occur in monerans. So commonly asexual method of reproduction occur in monerans. Mitotic spindle absent during cell division. Mitotic spindles are not formed in monerans, though this is the another characteristic feature, that is mitotic spindle is not formed in monerans. And last is, some of the monerans have the ability to fix nitrogen into useful nitrates. It means that some monerans are responsible for nitrogen fixation. They fix atmospheric nitrogen and convert that nitrogen into nitrate, and that nitrate is utilized by plants for their in the preparation of their food material. So especially in legume crops, root nodule is present, and in their root nodule a special type of bacterium, that is rhizobium, is present, that is rhizobium leguminosarum is present, and that rhizobium leguminosarum fix the atmospheric nitrogen and convert into nitrate, and that nitrate is utilized by plant in the preparation of food material. Now about bacterial cell, means bacteria. Now about bacteria. Bacteria is a primitive and unicellular organism, and bacteria are kept in under Monera. And in case of bacteria, the term bacteria was proposed by Ehrenberg in 1829. Who proposed the term bacteria? So the term bacteria was proposed by Ehrenberg in 1829. Now about occurrence. Bacteria are universal because they are present everywhere, in water, in soil, in air, inside body, in food material. So they are called universal because they are present everywhere. Some bacteria can survive in extreme range of temperature like up to 78 centigrade and minus 190 centigrade. In case of bacteria, some are useful and some are harmful for us because some bacteria are used in the preparation of different type of food material. In case of bakery industry, in case of beverage industry, and some bacteria caused different type of fatal diseases in case of animal body as well as in case of human body and in case of plant. So bacteria are a type of primitive and unicellular organism kept in under Monera, and now here we discuss about their characteristic feature, their useful and harmful activities, about their shape and sizes, and after that about reproduction in case of bacteria. Now about characteristics of bacteria. In case of characteristics, first number, bacteria are found in all kinds of habitats. All kinds of habitats means they are present in water, they are present in soil, they are present in food material, means everywhere on this earth. Bacteria are universal because they are present everywhere, so they are universal and present, are found in all kinds of habitats. After that, bacteria are prokaryotic organism. Prokaryotic organism means their nucleus is not well developed. Second thing, no any cell organelles like mitochondria, Golgi apparatus, after endoplasmic reticulum, means these all type of cell organelles are not present in bacteria, means inside bacterial cell no any cell organelles is present and their nucleus is in primitive stage because nuclear membrane is not present, means not well developed, so their DNA is naked because they are not surrounded by any nuclear membrane. Next thing, they are unicellular organism because made up of a single cell. After that, cell wall. Cell wall is present in bacterial cell, but their chemical composition is different from the cell wall of plant cell, fungal cell. Especially the cell wall of bacterial cell is made up of peptidoglycan. Peptidoglycan are murine. Peptidoglycan are murine, means cellulose is not present. Cellulose is not present in the cell wall of bacterial cell. After that, in bacterial cell there are two types of DNA are present, that is own DNA, that is bacterial DNA, as well as plasmids. Plasmid is a good vector used in the field of biotechnology because the self-replicating capacity is very high in case of plasmids. So in bacterial cell, extra chromosomal self-replicating DNA means the DNA which can replicate their number themselves. So self-replicating DNA segment is small part called plasmids occur in most of the bacteria, especially in case of E. coli. And the plasmids of E. coli used in the field of biotechnology like a vector because their replication capacity is very high, means they can replicate, E. coli can replicate their number within 20 minutes, means they can replicate, means after 20 minutes means their number will be double. Now about size and shapes of bacteria. So there are different type of bacteria are present on this earth, means variation also occur in case of the size and shapes of bacteria because all are not in equal size, all are not in equal shape. So due to variation their size and shapes are different. So in case of size, the bacteria ranges from 1 to 10 micrometer in length and from 0.7 to 1.5 micrometer in width. So width between 0.7 to 1.5 micrometer and length 1 to 10 micrometer. In case of shape, some bacteria are in oval shape structure, some are in spherical, Shape structure means in case of oval shape and in case of spherical shape without flagella, means flagella is not present in oval or spherical shape bacteria, and they are called coccus and plural is cocci, and due to absence of flagella they cannot move from one place to other place, means locomotion not occur in case of spherical or oval shaped bacteria. Some are in rod shape structure, especially they are called bacillus or bacilli. After that, some are in spiral shape structure, that is spirillum, plural is spirilla, and some are in C or comma shaped structure, they are called vibrio, and vibrio caused that is one example is there, vibrio cholerae. Vibrio cholerae caused cholera disease. So there are different type of shapes in case of bacterial cells. Some are in oval or spherical, after some are in rod shape, spirillum or vibrio. Now this is the structure of a bacterial cell. In bacterial cell, mean their shapes are different. So here one bacterial cell, and in case of bacterial cell, first thing flagella is present, and due to the help of flagella locomotion occur in bacteria, means through flagella they can move from one place to other place, and flagella is made up of a special type of protein, that is flagellin. After that cell wall is present, and cell wall is made up of peptidoglycan. After cell wall another layer, that is cell membrane is present, and cell membrane is made up of lipid and protein. All the bacteria are microscopic, means we cannot observe about the structure of bacterial cell with naked eye, because they all are microscopic, means with the help of microscope we can observe about the structure of bacterial cell. So this is a microscopic structure. After that many hair like large parts are attached with the outer wall, that is fimbriae, and after that pili fimbriae helps the bacterial cell to attach with any surface, that is wall or soil surface. Pili helps in reproduction. After that cytoplasm is present inside bacterial cell, and in bacterial cell a well developed nucleus is absent, so on the place of nucleus nuclear DNA is present, because they are not surrounded by any nuclear membrane. After many ribosomes are present, ribosomes are called protein factory, so ribosome molecules are present because they are non-membranous structure, so ribosome molecules are present for protein synthesis. After that plasmid is a self-replicating another DNA present in bacterial cell, because bacterial cell contain two type of DNA. First is their own DNA, that is bacterial DNA. After that plasmid. So this is the structure of bacterial cell. Now about nutrition in bacteria. So commonly in case of bacteria there are two type of nutrition, that is the autotrophic nutrition and heterotrophic nutrition. Means bacteria perform both type of mode of nutrition. They perform autotrophic as well as heterotrophic. In case of autotrophic, some bacteria are able to synthesize their food themselves, and in case of heterotrophic, some bacteria are depend on another body for their food material, because they are unable to synthesize their own food. So we know that all the green plants are autotrophs, because in all green plant chlorophyll pigment is present, and due to the presence of chlorophyll pigment they trap the sunlight, and after that photosynthesis occur in case of green plant, and through photosynthesis they synthesize their food. In bacteria a special type of green sunlight trapping pigment is present, and that pigment is called bacteriochlorophyll. In case of photosynthetic bacteria, means in case of autotrophic, two processes are there, photosynthetic and chemosynthetic. In photosynthetic means with the help of light, In case of chemosynthetic with the help of some raw inorganic raw material. So in photosynthetic bacteria, in some bacteria a special type of green sunlight trapping pigment is present, and that pigment is called bacteriochlorophyll. Bacteriochlorophyll trap the green sunlight, and through that light the photosynthetic bacteria synthesize their own food. One unique characteristic is there in the photosynthesis. In bacteria, photosynthesis never release oxygen. In photosynthesis, commonly in case of plant, in photosynthesis plant body release oxygen, but in case of bacteria, photosynthesis not release oxygen. Photosynthesis not release oxygen. So in bacteria, photosynthesis not release oxygen, and photosynthesis occur through bacteriochlorophyll, because bacteriochlorophyll trap the green sunlight. In photosynthetic bacteria, they are commonly occur at the bottom of ponds and lotus. They are present at the bottom of ponds and lotus. Second thing, chemosynthetic. Chemosynthetic bacteria are those bacteria they can synthesize their food with the help of some inorganic raw materials. Mean they can synthesize their food without light, but in the presence of some inorganic raw materials, and through the help of inorganic raw material they synthesize their food, especially in case of example nitrifying bacteria like Nitrosomonas. Nitrifying bacteria is a type of chemosynthetic bacteria. And next, iron bacteria. Iron bacteria, best example Ferrobacillus ferroxidans. What is the iron bacteria? Ferrobacillus ferroxidans. And another sulfur oxidizing bacteria, Vegeatoa. All these examples are very important for all type of competitive examination. Nitrifying bacteria, chemosynthetic bacteria, Nitrosomonas. Another chemosynthetic, that is iron bacteria, Ferrobacillus ferroxidans, and another sulfur oxidizing bacteria, that is Vegeatoa. In heterotrophic, some bacteria are depend on other living body or non-living body for their food material. So in case of heterotrophic, saprophytes. Saprophytes means some bacteria depend on dead and decaying organic matter for their food material. So they are called saprotrophs, and their mode of nutrition is called saprophytic nutrition. Some bacteria depend on living body. In case of plant, in case of animal, like in case of parasites like Salmonella typhimurium. Salmonella typhimurium caused typhoid disease. So Salmonella typhimurium is a type of pathogen which cause typhoid disease, and it is like a parasite. In case of symbiont, some are present in case of legume crops, means nitrogen fixation bacteria, that is rhizobium. So in case of heterotrophic, some are saprophytes, means depend on dead and decaying organic matter. Some are parasites like Salmonella typhimurium, after that Vibrio cholerae, Mycobacterium tuberculosis. And symbiont means they present in the root nodule of the legume crop, that is Rhizobium leguminosarum. So in case of nutrition, two types is there, that is autotrophic and heterotrophic. Now about reproduction. Reproduction also occur in bacteria, and there are two types of reproduction occur in case of bacteria, that is the asexual reproduction, means asexual reproduction commonly occur in bacteria, while other is sexual reproduction. In asexual reproduction, commonly binary fission and endospore formation occur. Binary fission is a type of asexual reproduction in which a bacterial cell like lactobacillus divides into two equal daughter cells, while in case of endospore formation, many spores are present inside the cell. So both type of means both processes occurring under asexual reproduction, especially in case of bacteria. Next is sexual reproduction. Next is sexual reproduction. In sexual reproduction, there are the requirement of two bacterial cell. Means in sexual reproduction, it is biparental, so there are two bacterial cells are required. So in conjugation, two cells are present. One is male cell, that is donor cell, and another is female cell, that is recipient cell, in which male cell have F factor, that is fertility plasmid, and male cell attach with the wall of female cell and transfer their fertility plasmid into the recipient cell, that is female cell. In transformation, transformation is a sexual process in bacterial cell discovered by Griffith in 1928. In transformation, the segment of DNA from one cell transferred to another cell through liquid medium. So in transformation, the DNA segment transfer from one cell to another through liquid medium, while in third number transduction, in transduction DNA segment transfer from one cell to another cell through viruses. So these are the sexual method of reproduction in under bacteria.